Assalamualaikum warahmatullahi wabarakatuh, and good day to all students. Hi, it is good to see you again. My name is Dr. Babul Arena Uthman, and this is the second part of nitrogen metabolism. In this video, I will try to give explanation on some figures in the lecture notes regarding amino acid biosynthesis, amino acid breakdown, as well as urea cycle. Please make sure you have your lecture note with you while reviewing this video, and don't forget your pen. You know well amino acid, right? You should. You have learned about it even during cell biology lecture. It is simply a building block of protein and play a vital role in our life. Human body need 20 different amino acids to function well, and that 20 amino acids are divided into two groups, essential and non-essential. This grouping of amino acid to essential and non-essential are also the simplest way to classify amino acids, which based on the essentiality for mammals to get it from diet, or also based on the ability of mammals to synthesize. Essential amino acid are amino acid that our body could not synthesize on its own, and therefore it is essential for us to obtain it from diet. Essential to obtain from diet by eating it. There are 10 of amino acid in this group, but you might find that some other article would say 9 or even 8. That is also true. Because of histidine and arginine, they only become necessary for babies. Body should be able to synthesize these two, but are not enough during active development stage, and therefore these two fall under essential category. This is the reason why sometimes essential amino acid is said to be 8 or 9, but me prefer to put the 10 here, 10 amino acid in this category. On the other hand, the non-essential amino acid are amino acid that can be synthesized by the body. This our body can synthesize it, and therefore are not essential to be included in everyday diet. The human body needs all of these 20 amino acids in varying degrees to be healthy and fully functional. All these 20 have distinct chemical structure and are used for different roles, but their primary role is to build up protein. Okay, to build up protein. That's the protein building block. From only 20 amino acids, the body is able to generate many thousands of unique protein with different function. This is the illustration to show the overall precursors for those 20 amino acids. All precursors for amino acid biosynthesis derive from intermediates in glycolysis in the TCA cycle, the citric acid cycle or Krebs cycle, or in the pentose phosphate pathway. The useful way to organize the amino acid biosynthetic pathway is to group them into family based on their biosynthetic precursor. Therefore, here in total, we have 6 groups of precursors family. Pyruvate family, aspartate family, glutamate family, serine family, aromatic family, and the phosphoribosyl pyrophosphate or histidine family. Pyruvate or pyruvate family give rise to alanine, valine, leucine, and also isoleucine that sharing oxaloacetate as the precursor. Meanwhile, oxaloacetate give rise to aspartate, asparagine, methionine, threonine, and also lysine, together with the isoleucine. On the other hand, alpha-ketoglutarate or glutamate family here give rise to glutamate, glutamine, proline, and also arginine. The precursor 3-phosphoglycerate here or serine family, this one, give rise to serine, cysteine, and also glycine. Phosphoenol pyruvate and erythrosulfophosphate, or also known as the aromatic family precursor, give rise to tryptophan, phenylalanine, and also tyrosine. And last but not least, this ribose 5-phosphate or the phosphoribosyl pyrophosphate, this PRPP, give rise to histidine, or this also known as the histidine family. Here is the same information as in the figure I explained before to show the family group of precursor. Okay, this one. Okay, this is the precursor 1, 2, 3, 4, 5, 6. 6 precursor that give rise to all total the 20 amino acid. Okay, this one. And the one in blue color here is the essential amino acid, while the one in red is the non-essential amino acid. Okay, that is about the family precursor for amino acid biosynthesis. Overall, 20 amino acid, all from only 6 family precursor: pyruvate family, aspartate family, glutamate family, serine family, the phosphoenol pyruvate or the aromatic family, and also the ribose 5-phosphate family. In the biosynthesis process itself, there are 2 common features of amino acid biosynthesis. The first one is this one, transamination. The second one is 1-carbon transfer. Okay, let's see some example of transamination that catalyzed by aminotransferase or transaminase. Glutamate here is formed by the transfer of amino group from one amino acid to alpha ketoglutarate and then form glutamate. And the amylation of glutamate will give rise to glutamine, catalyzed by glutamine synthetase. Remember glutamine synthetase in nitrogen assimilation pathway? So this is the same enzyme. So thus for aspartate here and also asparagine, it's a transfer of amino group from one amino acid to oxaloacetate that will give rise to aspartate. And further reaction by asparagine synthetase give rise to asparagine. Alanine, on the other hand, here is from pyruvate. When pyruvate receive the amino group from another amino acid by aminotransferase. And for serine here, okay, is from 3-phosphoglycerate. The second reaction in serine here from 3-phosphohydroxypyruvate to form 3-phosphoserine is also a transamination process. Basically, transaminations involve an amino acid as the amino donor, this one, and a keto acid as an amino acceptor. This amino donor, okay, is normally another amino acid, especially amino acids that carry 2 amino groups such as glutamine and also glutamate. Glutamate known as the universal amino donor. Okay. On the other hand, a keto acid is a compound with oxo group, this one. Okay, that can be the acceptor for amino group. One common keto acid is alpha ketoglutarate. Okay, so transamination is basically a transfer of amino group from the amino donor, okay, to a keto acid to form a new amino acid here, a new product catalyzed by transamination or transaminase or aminotransferase. The second most common amino acid biosynthesis is 1-carbon transfer. For example, here is the formations of amino acid glycine from serine. Okay, the 1-carbon acceptor is this tetrahydrofolate, and it becomes the carrier of the 1-carbon group from serine that catalyzed by a reversible reactions by serine hydroxymethylase. Okay, so this 1 carbon from serine is transferred to tetrahydrofolate. Tetrahydrofolate become the carrier, and then this 3-carbon serine okay, form the 2-carbon glycine after it transfer of its 1-carbon compound here to tetrahydrofolate. Some pathway of amino acid biosynthesis are simple, others are not. Others can be very complex. Okay, those I showed earlier are all the non-essential amino acid biosynthesis here. The synthesis of alanine, aspartate, glutamine, all these are the non-essential. They have more simple pathway, just through aminotransferase and maybe 1-carbon transfer. But the biosynthesis of essential amino acid here are normally far more complex, such as the synthesis of lysine, methionine, threonine from the precursor of aspartate family. And another thing here in amino acid biosynthesis pathway, okay, the pathway for the same amino acid are not always the same to every organism. For instance, the biosynthesis of tyrosine here in microbes and in mammals are completely different. In mammals, the biosynthesis of tyrosine is pretty much simple. it is synthesized from phenylalanine, catalyzed by phenylalanine hydroxylase. But in microbes, the tyrosine and phenylalanine are synthesized from phosphoenol pyruvate and erythrosyl 4-phosphate through a more complex pathway. Now take a look here. This is the information I extracted from the previous slide. Okay, the precursor and the amino acid, and also the synthesis of tyrosine by mammals. Okay, mammals synthesis tyrosine from phenylalanine through hydroxylation process catalyzed by phenylalanine hydroxylase. Tyrosine in fact a non-essential amino acid. Tyrosine is a non-essential amino acid. On the other hand, phenylalanine is the essential amino acid. What does this mean? Okay, it means that in order to synthesize a non-essential amino acid of tyrosine, an essential amino acid phenylalanine is required. Therefore, the synthesis of tyrosine here is depending on the diet intake of phenylalanine. This brings to another scenario. PKU or phenylketonuria. Phenylketonuria is a genetic disorder disease due to the dysfunctions of phenylalanine hydroxylase. So in PKU, the phenylalanine hydroxylase is dysfunctioned, and this will then cause accumulations of phenylalanine in blood, as phenylalanine could not be hydrolyzed to form tyrosine. So now in PKU patient, the tyrosine, okay, this one tyrosine, is now become the essential amino acid. Essential amino acid, okay, because body failed to synthesize tyrosine. Okay, tyrosine is an important amino acid, especially for synthesis melanin. Okay, remember the transamination process from the previous slides? I would like to introduce you to a molecule that play an important role for this to happen, named as pyridoxal phosphate. Pyridoxal phosphate plays a major role in most amino acid biosynthesis. This pyridoxal phosphate is basically the biological active form of vitamin B6, and it participates in the catalysis of a wide variety of reactions of amino acid, not only transamination but also decarboxylation. But here I'm going to show you its role in transamination. Transaminations involve one amino acid and one keto acid as a substrate. Okay, this one as S1, S2, S1, S2, as well as one amino acid and one keto acid as the product. Here P1, P2, P1, P2. The pyridoxal phosphate bound to enzyme that catalyze amino acid biosynthesis and form a coenzyme, this one. This coenzyme of pyridoxal phosphate then bound to the first substrate, that is the amino acid that act as the amino donor, and form this imine or a sheaf base with the alpha amino group of that amino acid. Once that happens, rearrangement occur and hydrolysis of this isomeric imine give a keto acid that is the first product and also a pyridoxal coenzyme. This pyridoxyamine coenzyme will then bound to the second substrate S2 here, i.e. that is the keto acid, and another rearrangement occurs similar to the first one, and the hydrolysis of this imine give rise to the second product P2 here, that is the new amino acid. All reactions here are reversible, and this is how actually amino acid pass its amino group to a keto acid to produce a new amino acid required by the system. All due to this role of pyridoxal phosphate. So, done on the biosynthesis. Let's move on to the next part. It is time to break it down. This figure shows the catabolites of that 20 amino acid. That 20 amino acid that we learned earlier, in their catabolism, they will be degraded to just 7 metabolites. That are this one, pyruvate, oxaloacetate, fumarate, succinyl-CoA, alpha-ketoglutarate, acetyl-CoA, and also acetoacetyl-CoA. Out of these 7, 3 of them are the similar metabolite of their precursor. That are this one, the pyruvate, oxaloacetate, and also alpha-ketoglutarate. Okay, but yet the another 4 is still a very common metabolite that you may found in glycolysis, the citric acid cycle, the Krebs cycle here, as well as the pentose phosphate pathway. In amino acid catabolism, we can categorize all that amino acid into 2 groups depending on the fate of the end product. They can either be glucogenic or ketogenic amino acid. What does it mean by glucogenic amino acid? Okay. Glucogenic amino acid, okay, this one, glucogenic amino acid is amino acid whose carbon skeleton is degraded to any 5 of the degradation metabolite. Either it degraded to pyruvate, oxaloacetate, fumarate, succinyl-CoA, or alpha-ketoglutarate. Okay, it is called glucogenic because all of these 5, pyruvate, oxaloacetate, fumarate, succinyl-CoA, and alpha-ketoglutarate, could then be converted to glucose through gluconeogenesis. Okay. Ketogenic amino acid, on the other hand, is amino acid whose carbon skeleton is degraded to either acetyl-CoA or acetoacetyl-CoA. It is called ketogenic amino acid, okay, because the degradation of that amino acid lead to the formations of ketone body. This one, thus, could not be used to form glucose, but instead can be used to form other molecules such as lipid. As you can see here in purple color, many amino acid can also be in both group. That is, can be glucogenic and also ketogenic as the degradation of that amino acid, okay, such as tryptophan, tyrosine, give rise to more than one metabolites. For instance here, tryptophan can also be degraded into acetyl-CoA. Tryptophan also can be degraded to pyruvate. So they can be both glucogenic and also ketogenic. Anyway, it is clear here that only lysine and leucine are the true ketogenic amino acid. So how is actually amino acid is broken down? One most common step before amino acid is broken down is that it transfers the amino group to alpha-ketoglutarate to produce glutamate. Okay, glutamate is the universal donor. Okay, it is readily give up or release its amino group either for the synthesis of other amino acid or to release free ammonia. In amino acid catabolism, the amino group in glutamate will be deaminated, okay, through glutamate dehydrogenase or GDH here, GDH, and release free ammonia. Alternative to the deamination process, oxidation reaction may also contribute to the amino acid catabolism. For instance here, the oxidation process of glycine by glycine oxidase to form glyoxylate. Another pathway is a direct deamination process by serine deaminase as well as by glutaminase that release free ammonia from serine and glutamine respectively. So here, as summary, degradation of amino acid could proceed through several pathways, which are either by deamination through GDH, glutamate dehydrogenase, serine deaminase, or glutaminase, or by oxidation such as by glycine oxidase. All of it will release free ammonia. This one, okay? This free ammonia is toxic, and even small amount will damage the nervous system. Ammonium intoxication can also be lethal. So, what should be done to this free ammonia to prevent intoxication? The amounts of free ammonia that are present resulted from amino acid catabolism can be regulated through urea cycle. Okay, this urea cycle is basically a formation of organic nitrogen as a waste product. In human and most other terrestrial mammals, urea is the major nitrogen excretion product. Why? Because urea has the advantages of being relatively inexpensive. Okay, it's inexpensive to produce. Not much energy is used to produce urea. It also soluble in water and non-toxic. On the other hand, aquatic animal excrete ammonia in the form of ammonia itself, while birds and reptiles excrete ammonia as uric acid. Here, we're going to focus on the excretions of excess free ammonia from amino acid catabolism through urea cycle. Although most tissues can synthesize urea, most urea is produced in the liver. Okay, in the liver. Excess nitrogen from muscle Okay, excess nitrogen from muscle is transported to liver as alanine, while nitrogen from other tissues are transported to liver as their respective amino acid. Let's first take a look here on the overall reactions of urea cycle. Urea, this one, consists of two amide group, okay, 1 and 2, that bound to a carbonyl group. One of these amide group originated from the free ammonia, the one in green color here. This is the free ammonia from amino acid catabolism. And another amide group here is contributed by the amino group from amino acid aspartate. The carbonyl here okay, is from the carbonated iron that readily available in system, and the byproduct of urea cycle is fumarate, that is the remaining carbon skeleton of aspartate. This is the illustration to show the overall process of urea cycle. On the upper part here is some of the pathway on amino acid catabolism that release free ammonia to the system. Here is the release of ammonia from glutamine by glutaminase. Okay, it releases free ammonia. And here is how alanine from muscle is transported to the liver and transfer its amino group to form glutamate, which will then, this glutamate will be degraded by glutamate dehydrogenase to form free ammonia. So this is the free ammonia that need to be regulated and excreted out through urea cycle. Okay, and the remaining figure here is the urea cycle. The urea cycle requires 5 reactions in total. Okay, 1, 2, 3, 4, 5. With 4 actual reactions of the cycle, this one, 1, 2, 3, 4, and 1 is outside the cycle. The first reaction here is catalyzed by carboxyl phosphate synthetase 1 or CPS1. CPS1. CPS1 is the mitochondrial enzyme that catalyzes the condensation steps of ammonia, this free ammonia, and carbonate to form carboxyl phosphate. This carboxyl phosphate will then enter the urea cycle. The cycle begins with the additions of carboxyl phosphate to ornithine by ornithine transcarbamylase. This is the first urea cycle enzyme, ornithine transcarbamylase, to produce citrulline. Citrulline then leaves the mitochondria using a specific transporter because the remaining reaction occur in the cytosol. Once in the cytosol, citrulline is combined with aspartate here, okay, by arginosuccinate synthetase to form arginosuccinate in a reaction that requires ATP and produce pyrophosphate and also AMP. The aspartate here will become the second amide donor for the formations of urea, the green one here. The blue one is from the free ammonia from amino acid degradation. The next enzyme is arginosuccinase. Perform a cleavage reactions of arginosuccinate that release fumarate and the amino acid arginine. Note that here that this arginine contains nitrogen from the free ammonia, this one, and also nitrogen from the aspartate. And the remaining carbon skeleton of aspartate is released as fumarate. Arginine is then cleaved by arginase, the last enzyme here, to release urea and to regenerate ornithine. Ornithine also has a specific transporter that allows it to re-enter mitochondria and then completing the cycle again. So yes, that is the urea cycle. Now let's see how this cycle is regulated. The urea cycle is regulated by 2 factors. One is by substrate availability, and the second one is by the enzyme carboxyl phosphate synthetase 1. CPS1 requires the presence of allosteric activator N-acetylglutamate. This N-acetylglutamate derived from the acetylations of amino acid glutamate. So basically, amino acid glutamate here is not only the source for free ammonia, but also the source for the activator for CPS1. This regulation also means that CPS1 here is the rate-limiting enzyme of urea cycle. Other enzymes of urea cycle are controlled by the availability of the substrate. The higher the substrate, the higher the reaction. Fumarate released from urea cycle have several fates, including it will re-enter the urea cycle as aspartate. Again, this is through fumarate malate oxaloacetate intermediate. The detail of this reaction you can see here. Okay, this is actually the urea cycle, and when urea cycle release fumarate, and then this fumarate can re-enter the urea cycle in the form of aspartate, the second amino donor, by the transformation through malate and oxaloacetate, and then back to aspartate. Fumarate can also enter the TCA cycle here as TCA intermediate, fumarate, malate, or even oxaloacetate. Well, as you can see here, the urea cycle and the Krebs cycle are interconnected by this fumarate. The urea cycle is actually the first of the two major metabolic cycles discovered by Hans Krebs. Yes, it is the same person that discovered the Krebs cycle. Okay, in fact, the urea cycle was the first biological cycle to be discovered and helped establish the concept of the discovery of TCA cycle or the Krebs cycle. That is why sometimes these two are called Krebs bicycle, as they are interconnected to each other through fumarate. Interesting, right? So, that is all on the second part of nitrogen metabolism. If you have any further question, please do not hesitate to drop an email to my email address here, ariana@ukm.edu.my. Thank you very much and wish you best of luck.